Greetings, everyone. So I'm very happy to be here giving a talk and sharing my learnings on this wonderful seminar series that has been organized by the Gangwal School of Medicine, Science and Technology, IIT Kanpur. So when I got this invitation, I was reminded of this quote that I had read by a researcher last year. And this is very well relevant today. And so I feel that this is a very thoughtfully crafted series because for sure, pregnant women are often left out of research. So as a cancer biologist, I've been working in the field of epigenetics of cancers for many years now, particularly in the tumor microenvironment. And also as a teacher of a master's program, I do know that transgenerational transmission of traits or intergenerational transmission of traits is the most fascinating, yet understudied, and perhaps the most important in clinical and biomedical research. So one aspect of that is microchimerism, which I will be talking about today. Anecdotally, pregnant women have always been advised to be careful about what they're eating, the habits, as it may influence the fetal health and growing child in many ways. So the mother has always played the role of a nurturer and has always been the one who has given to the child and the growing fetus. So we also know that many of these aspects, such as nutrition, can influence the outcome or the molecular repertoire and phenotype and trait that we observe. And that's because these are acting as a regulatory layer over and above the genetic sequences that we are born with. Triggers from the environment or the nutrient, they influence the cellular function and the molecular expression in such a way that it changes the trait or the observed phenotype. And this is what happens, and these modifications or these epigenetic modifications are happening in the histones, the non-coding RNA, the DNA methylations, the microbiome, and affect the germ cells such as the sperm and the egg, and which then, when they combine and come together in the progeny, are able to offer a regulatory role or a regulatory control which is over and above the genetic sequences that the particular progeny is born with. Epigenetic changes can be transitional or may be required to be retained over a much longer period of time. So a confluence of all of these epigenetic changes are required to bring about this complex information which is gathered and memorized by the cell and stored in the genome. So, we have also been working in this field, although in cancer biology, where we look at how the tumor microenvironment influences all of these different epigenetic phenomena, and some of the published work is mentioned here. Such long-term inheritance patterns are particularly relevant for placental mammals, and many of the studies that have been done to look into subsequent generation have been on mice, and also some examples where humans were studied, such as that of the Dutch famine of 1940s, where how the resources, nutritional resources, were adapted to by subsequent generation was analyzed. So in this case, if this is the parent's nutrition or epigen or any other stressors would influence the F1 generation, which is the fetus. But since the fetus also has the germ cells which have been developed, then that means that ultimately the outcome of changes in these germ cells would be seen in the F2 generation. And once the F2 generation is carrying these changes and they are giving rise to the next generation, it would become transgenerational, while all of these others would come under the intergenerational passage of information. So turns out that that passage of information is not just one-sided, and pregnancy is a two-way communication. And the information which transferred uh, is transferred not just from the mother to the fetus, but also from the growing fetus to the mother. And this has been analyzed because traces of genetic material that were previously not found in an individual were seen after the pregnancy. So this is considered as an epigenetic phenomena because you have a chimeric individual which is formed and likely to be influenced by genetic material that does not belong to it. So we know of chimeras as being composed of two different types of genetic material. Well, in this case, you have the fetal microchimerism that we are studying, wherein the fetal DNA or the fetal cells are transferred onto the mother. And the fetus can also impact the mother in many ways, as we'll see in the next slides. Initial studies carried almost 250 years ago demonstrated that the fetal and the maternal circulations were separate. So we started off with placenta being recognized as a barrier, and from there on, from looking at the first report of fetal cells being found in women to transplacental transmission of erythrocytes and many other fetal cells thereon in the 1990s, we have now come to recognizing placenta as a kind of a connectome because of the way that the information is transmitted across it. These fetal-maternal microchimeric cells have been shown to be deposited and settled in different parts of the mother's body and different organs, and their role is still debatable. Research is coming in which some of it shows that it has a positive impact on the maternal health and some says negative or even no impact. What we do know is that the transfer itself is asymmetric and many more fetal cells are transferred to the mother than vice versa. What has also been observed is that you can see some of these cells in the mother's body as late as after 27 years of the last pregnancy. These microchimeric cells and their exchange can be studied as an inheritance pattern, as can be seen over here, that mostly it's a bidirectional microchimerism where the exchange happens between the fetus as well as the mother cell. However, in case of twin microchimerism, the exchange also takes place between the two twins shown by the purple dot here, with the second twin which is passing through to the first twin as well as to the mother. In case of multi-generational microchimerism, things become a whole lot more complicated, and this is better explained in the pedigree chart that I will show you. So here you can see that this is a pedigree which is showing 3 generations, and in all of these pedigree analysis, the males are shown in squares and the female in circles. So here is the mother who has given rise to 4 progenies in the second generation. And the mother, as you can see with the color code, is carrying the fetal cells from all of the progenies that she has had. The interesting part is that if you look at the progenies, the first progeny only carries cells from the mother, but the second and the third are also carrying, and the fourth are also carrying the fetal cells of their elder sibs. And in this case, what has been shown is that this last progeny of the second generation is also carrying the fetal cells of her own kids. So in this case, especially it's pertinent to note that even though she had a miscarriage, and she would still carry the fetal cells or the cell-free DNA of the daughter who was aborted or miscarried. So this is the brown dot shown here. So what are these different types of cells which are passed from the fetus to the mother? Well, a population of them are the pregnancy-associated progenitor cells, or also the fetal maternal stem cells, and these carry stem cell-like properties. You also have the fully differentiated fetal cells such as trophoblasts, blood cells, erythrocytes, which are also lymphocytes, and erythrocytes, which are also passed on. Important to note that the cell-free DNA also exists, so it's called cell-free fetal DNA, SFF DNA, and their presence was first identified in the 1990s. And all of these cells, as well as the cfDNA, are important for non-invasive prenatal diagnosis. It has been determined that the highest number of fetal-maternal microchimeric cells peak at different times in different animals, and the highest number of fetal-maternal microchimeric cells in the humans can be seen at 36 weeks of gestation, and thereon most of the cells are quickly destroyed. However, they are still known to persist till very long. The 27 years written over here is because of that 1996 study that I spoke about. So landmark discovery was made in this 1996 study where the fetal cells and the cell-free fetal DNA was detected for the first time with the help of flow cytometer, and as well as PCR specific for Y chromosome. So the male fetus was fetal microchimeric cells were detected, which makes it easier to get some specificity, because if it was a female child, it would be difficult for researchers to detect the mother's XX from the female child's XX, and in this case, the Y chromosome can be detected. So the cohort had 32 pregnant women who were pregnant at the time of the study, and 8 non-pregnant women who had previously given birth to male child 6 months to 27 years earlier. So blood was drawn and cells were sorted using the flow cytometer and several CD markers, which included 34 and CD38. And then from these sorted cells, DNA was isolated, and with the help of quantitative PCR, the Y chromosome specific gene was detected. So there were many aspects to the study, but the most interesting was that of the results of studies on women who were not pregnant at the time of blood sampling. And here you can see that in this table, particularly this patient marked as patient 5, that the previous pregnancy was 27 years prior to the blood sampling, and there was a detection of the Y chromosome gene in the lymphoid progenitor cells, which are isolated from this particular participant, indicating that indeed there is persistence of the fetal DNA for years after the pregnancy, and also persistence of previous pregnancies' fetal progenitor cells, as was observed as in many of the women who were analyzed under this cohort. So these cell-free fetal DNA could also be detected using PCR for Y chromosome in Alzheimer's disease patients compared to the normal, and many brain regions were obtained from each of these subjects, and then quantitative PCR was done after determining that a fetal cell was indeed present. This confirmation was done using fluorescence in situ hybridization, where the cells can be seen as being a male fetal cell. Then quantitative PCR was performed, and it was seen that there was a distribution of the fetal cells and hence the DNA in different regions of the brain. So with this study, the authors concluded that fetal microchimerism could be seen in multiple regions of the brain, and that less microchimerism was seen in Alzheimer's disease patients compared to the control, pointing to a protective effect of the fetal fetal cells in the control. With developing technologies, the current work in this budding field has started working on acquisition of these cells with greater sensitivity. So in this 2024 paper shows a micro nanochip which has been fabricated and onto which CD147 was layered to capture the fetal nucleated red blood cells specifically. The authors then followed it up with addition of the metalloprotease MMP9, which results in a dislodging of these cells. The idea is to capture these cells and thereby enriching them and using them for many downstream next-gen sequencing, copy number variation, and other studies. The characterization of the cell is done using specific markers. In this case, for example, HbF, which is the hemoglobin F specific for the nucleated RBCs. You can see that because it is nucleated, the DAPI is staining both the nucleated RBCs from the fetal source as well as the WBCs from the mothers. And such technologies have therefore greatly expanded the scope not only of prenatal diagnosis but also for understanding the biology of these fetal cells. So once these DNA or cells are acquired, they can undergo the targeted sequencing or random sequencing and a number of tools that are available. And it's pertinent to point out that the false negative and false positive in prenatal non-invasive diagnosis is very important, especially when you're comparing it with the maternal cells. So many of these detection techniques have been utilized in various studies to understand the fetal-maternal cells. This is a 2009 paper where virgin mice were mated with GFP-expressing male mice in both heterozygous and homozygous conditions. The cells that were isolated from various tissues postpartum were analyzed on a flow cytometer for their fluorescence, and indeed fluorescence was seen to be occurring in many of these cells, and more so where the GFP-expressing male mice were homozygous. It was also seen that from a panel of markers for various biological points such as migration, stemness, angiogenesis, many of these markers were expressed in different tissues, indicating that, and this was done in specific cells which are isolated, which were showing the GFP expression, so done through a flow cytometry. So indeed, these cells housed in various tissues had specific patterns of expression of many of these markers also. It was important to note that these fetal stem cells, and particularly fetal mesenchymal stem cells, were responsive to cues from the mother, and this allowed them to differentiate into different tissue types. And this was observed from the study where they could isolate these fetal stem cells and then allow them to be differentiated into adipocytes and even into bone cells. So the oil red stain here stains the lipids, and this was an indicator of the adipocyte differentiation. Also, it was seen that these microchimeric fetal cells played a huge role in, or played at least, if not a huge, a significant role in maternal wound healing after pregnancy. So in this panel here, this is showing a cesarean scar, and it was seen that at these positions, these cells would, which showed the Y chromosome lighting up through the fluorescence in situ hybridization, were actually housed near to the scar at the time of its healing. Particularly, these fetal derived cells were also able to express markers such as the cytokeratin marker here, showing that they were present at the time of the healing of the tissue. So these indicated that these cells were indeed responsive to the cues that were coming from the mother, and in this particular case also playing a role in wound healing. So researchers were curious to know on how the fetal cells would behave when the maternal cells, when the mother acquired melanomas during the gestation period. So for this, they took mice and they mated to GFP expressing males, and once pregnant, these pregnant mice were actually injected subcutaneously with melanoma cells, and the tumors were allowed to grow. And then these tumor cells were analyzed for the presence of the fetal cells and as well as some other markers. So in this case, in these two panels, it was seen particularly that at the site of where the melanomas were growing, the fetal cells homed in onto those sites during the gestation period. And the last panel shows the collected melanoma lesions from human patients, from women who were pregnant. And again, it was seen the green dots are showing the Y chromosomes, indicating that the fetal cells had homed onto the melanoma site. Retrospective analysis was done with paraffin-embedded surgical samples, and here they could see that male fetal cells with the Y chromosome co-localized with the CD31 marker of the tumor, again indicating that even in retrospective samples, the male fetal cells could be identified as present closer to the tumor. Ultimately, they concluded also that the frequency of the fetal microchimerism in melanoma in pregnant women was much more as compared to the benign. So accumulation of these fetal-maternal stem cells has also been seen with other disease pathogenesis such as the cardiac. And in this case, a study was done where the male AGSP mice was crossed with the virgin female, and the pregnant mice were actually induced with myocardial infarction. And it was seen that so some of these pregnant mice, those which were containing about 50% of EGSP fetuses, were analyzed for their heart in terms of the EGFP glowing cells, indicative of the fetal chimerism. So in this case, what was seen was that in the injured myocardial infarction-induced mice, the percentage of GFP cells that were accumulated in the heart were much larger than that in the control, and this was seen in 1 week and as well as in 2 weeks post the injury. The heart cross-sections were also analyzed for their content, and these EGFP glowing cells can be seen at the site of the injury, and this is in 3 weeks and 4 weeks. And what was also noticed was that there was significantly larger number of cells that accumulated in the injured or the border area with the injury as compared to the uninjured area. And this basically proved yet again that the fetal-maternal stem cells happen to accumulate at the site of an injury. So with the premise and the knowledge that these fetal microchimeric cells could be useful in wound healing, a set of researchers looked at the sickle cell disease and particularly the symptom of leg ulcers, which is seen in many such patients. They used a mouse model initially to determine that the postpartum mice had better wound healing as compared to the virgin, and this can also be shown here where the wound area is much lesser from day 3 to day 5. They isolated these fetal cells and performed transcriptomics and observed that the markers of associated with endothelial cells and leukocytes, which are representative of migration and angiogenesis, both processes, associated with your wound healing were observed. Then they picked a retrospective cohort of single cell disease patients, and they went about analyzing whether what the burden of the leg ulcers was in these women, and they found that those single cell disease affected women who had ever had a baby at any point of time were significantly lesser, were showing significantly lesser leg ulcers as symptoms as compared to the nulliparous women. So this study again highlighted the role of the fetal microchimeric cells in wound healing in the maternal tissue. Apart from the few of these cases that I have mentioned, there are many other impacts of the fetal-maternal microchimerism on different disease pathogenesis, with such as eclampsia, as well as rheumatoid arthritis, lung epithelial cells, and including the contradictory reports in breast cancer. And all of these point to the importance of studying the fetal-maternal microchimerism in more detail. So we discussed three instances where there are many others which I have not put in here, that where the fetal-maternal cells help the mother in a variety of situations, especially pertaining to lesions or injuries. So the pertinent question becomes on why it would do it. And from the evolutionary perspective, we know that there is a motive or a reason behind every event. So we could think about whether it is in the interest of the survival of the fetus to actually help the mother during the gestation, and that is what is driving this connectome which has emerged in the placenta. So I hope I have managed to convince you that these fetal cells which are traveling to the mother are very important for the maternal health, and looking forward to more data coming in from the single cell genomics, which is currently ongoing, and spatial transcriptomics, who are studying this mother-fetus interface in more detail. So thank you all for watching, and thank you to the organizers for giving this opportunity.